So welcome all of you for the 01 session on the topic entitled as the basic cell and membrane biology. So my very first question in general, do you have any idea about the cell biology in general at the master's level, or maybe any of you means just, if not, then yes, what we are going to cover in this entitled 01 subject is here are the subsections of the title, which we will be trying to cover all through the 1 week of this lecture series. And we have a wonderful group of speakers also, both from the campus and out of the campus. So which will try to imbibe a kind of to how to approach and learn the basic understanding of the membrane and the cell biology. So our first lecture, although it could have been on third list, but since our speaker was out of the station, our external speaker, so we have reoriented the session, and the organelle communication and membrane traffic has been on the first day, which I will be trying to cover. And our next topic is going to be by Dr. Saurabh from Vizag. He's an assistant professor and PhD from CCMB. So he will try to explain you more from the biophysical perspective of the membrane organization and dynamics, followed up by lecture by the lipid protein interaction, the membrane protein organization. And then we will cover the aspects on the membrane remodeling. So how the membrane remodeling is going to be involved, both on the biophysical aspects and is there any kind of a physiological relevance in some human health as well as in the disease system. So that part will be covered by our colleague, Dr. Lokendra Sharma. And follow-up lecture will be on, whenever there's an infection mediated by the viruses, how the viruses utilize the membrane or manipulate the membranes to hijack the host cell as well as the membrane pathways. And then not only the pathogens, but our cellular system, our body, or different organs, they also undergo the process of fusion. So what are the kind of membrane manipulations taking place during the development and the disease condition? We will try to shed light on that. And then we have a wonderful speaker in our last session where we will be really trying to cover the cell fusion and its implication in the health and the disease, which will be covered by Amita Agrawal from Clinical Immunology Department and Dr. Amrit Gupta. So both will try to cover the different aspect of these cell fusion models. Probably one will be going on the osteoclast aspect, and the next would be on the placenta biology aspects. So let's try to have the understanding of the organelle and membrane trafficking events. So I know since your childhood days, you might, it's not a childhood, but in fact, the high school literature aspect, everybody of you know organelle. Oops, slides are not moving very soon. Yeah. So this stationary images, all of you might have seen, our membrane is our cell as compartmentalized. It's delimited by the membrane, which you see as a plasma membrane. But beyond that, the cells are also limited by the intracellular compartmentalization. And these intracellular compartmentalization have a number of functional and role. And they have been mentioned as a mitochondria, nucleus, cytoplasm, lysosome, Golgi, networking, and a number of the other emanating bodies which are going to be secreted and shut off over the period of a time. So these are all organelles, and we know they do perform a different function, as if our nuclear membrane, which is the sole function to follow the central dogma to have the DNA as a material where they are going to be translated outside in the cytosol from DNA to RNA, and then proteins are being synthesized. The proteins synthesized mainly in the ER are being then trafficked to the different destination, which we will try to cover in the membrane trafficking aspect. How are they going to be different trafficking events are going to regulate the process? So those are all aspects are going to be how the ER where the proteins are going to be synthesized. They are being delivered to the different destination site. They are going to act as a kind of like say the delivery system. That's what we will try to cover in the membrane trafficking aspects. And then in certain cell type, because in the course of the evolution, so vacuole or osmoregulatory systems are also being involved. So certain vacuole systems are being there. They have a functional regulation, the osmotic balance. And those are like say how they get disturbed in certain disease conditions means the vacuole itself is an entity. The mitochondria, we know it's like say the main source for the energy, which produces energy and bioenergy and which can be utilized for the number of the different events. And then we have like say the lysosome where they are going to degrade our misinformed proteins, the cell organelles, by the different related process known as the autophagy. And I think probably most of you have already know about it. And then the dynamics on which, like say, the platform where they are going to be mobile, the framework is being provided by the microfilament structure where the organelles they traffic across the cellular system. So we know, like at least from this image, it looks like apparently, oh, all the organelles are really stationary, and that means. But what we are trying to cover in this session of the series is of the cell biology and membrane biology is a basic understanding and how the cell biology is developing and growing in this field. We will try to shed light on those aspects. So at least to give a glimpse, like say where we are moving to understand the biology of the both at the intraorganal, and the intracellular level, as well as the membrane biology level, and both from the cell biology and the biophysical aspects perspective. So you will see in this video, like our organelles are not stationary, rather they are like say highly dynamic. So you see, like say we have a different organelles labeled with the different dyes or proteins. We will get back to it later. But you see the organelles are highly dynamic in nature, in space and time. Be it a peroxisome, be it a mitochondria. Yeah. Hello. Yeah.
It's not visible.
It's not visible.
No, sir. The slide of the video is not there.
But it's working in my laptop. I don't know why.
We could see the folder, like demo with watermark visualization.
Want to say that the organelles are highly dynamic in nature, both in the space and time. So, and that allows the cells to interact with the different organelle level. Be that, like say, what I mean by the organelle level is like say, means ER is going to communicate with the nuclei, mitochondria, and different organelles, like all the organelles you can imagine, they are all going to be highly dynamic in nature and they will cross-talk with each other. So that gives, like say, the kind of an inception, like to perform the specific function or signaling aspect, organelle at the the cell at the intracellular level need to be highly dynamic in nature to perform the different function of the cellular system. And that's what we will try to cover in this, that how the organelles, they are going to also interact with each other. So, but before going into that, I think you people have already covered this in the different series of the lectures. I'm not going to that, but we can see our eyes have a resolution with something to resolve the two closure points. So every imaging approaches have a different resolution limits. Our eye can resolve up to the lowest level of like say 1 or 100 micron, even less than that. So similarly, like say that as the microscopy has evolved, we are able to try to go deeper and deeper so as to resolve, like say, to achieve the better resolution. So electron microscopy has been a classical approach to visualize, like say, the viruses, which it falls out to the closer to some range of like say 100 nanometers or lesser than that. And even like say means we can fall to the level of the atom levels. But there's a limitation for these studies, like say you have to, the membranes, they get, you get an artifact with that. It's, you have to fix the cells and there are a lot of artifacts. So you will not be able to capture the structure in its own, like say, means the native form or conformation or the dynamics. So, and that's where we limit, like say, to means utilize these kind of an approaches in every conditions. So over the period of time means different approaches of fluorescence imaging has also been involved, means developed, and we have a confocal image microscopy where you tag the specific proteins or specific lipids with the tag, and then you try to, like say, localize your protein of interest. But simply, like say, in a simple inverted microscope, if you express a GFP, means it's not going to, like say, localize a particular organ or organelle, I would say, organelle at a time. It's going to, like say, fluoresce everywhere. So that's the one limitation with the like confocal or those things. So down the lane, like say, if you want to really, like say, pinpoint the specific organelles, like say ER, Golgi, nuclei, you need to go like to achieve, like say, better resolution. So both the tool, I would say both the tool, I mean, here comes the tool is like say the microscopy, as well as like say the fluorophore. Because even different fluorophores are also going to behave differently in a different contextual manner. So with that perspective in a mindset, like to what a kind of a resolution you are going to achieve to target, like so identify or see the dynamics of the different specific organelles, it's very important to see, first of all, like say, right selection of the microscopy tool. That's the one thing. And then accordingly to complement it with the different fluorophore analogs. So depending on a series of the different super resolution microscopy has been developed, I think you people have better understanding because the courses are similar courses you may have gone through with that. So here, like, as I mentioned, definitely a selection of the microscopy is one aspect, but at the, not at the cellular level, but if you go deep inside, like say at the organelle level, ER level, mitochondria level, plasma membrane level, you need to label it with the precise fluorophore molecule. And number of the different approaches are now, like say, in the past decades has been developing to achieve, like say, to have a better resolution. Because if there is, like say, even if you use the fluorophore and if there is, like say, the overlap with, like say, the different spectral overlap, there's a crosstalk, and you will not be able to, like say, identify or, like say, you will misinterpret your data. That's what I need to say, because those are the limitations with the microscopy. Your fluorophore should be, like say, very precise and, like say, in terms of, like say, the excitation and the emission, and they should not overlap with that. Otherwise, like the false positive are the like main concern with the microscopy images. So yes, like conventionally, like say, different cell permeable different fluorophores are already commercially available, like means depending upon the nature of the mitochondrial membrane potential dyes are there, yeah, mitosoxes are there, and similarly, like say, the lysosomes which are traditional low pH. So the low pH sensors are there, and these are really helps, like say, in some way, like say, for the certain pathogens when they just try to take a route or infect your host cellular system. We will see in subsequent lectures whether it goes undergo by the plasma membrane infection pathways or by the endosomal pathways. And when it's going to be the endosomal pathways, they will definitely try to fuse with the lysosome. And then if you have, like say, means your virus or the bacteria labeled with the different fluorescent molecules, and then you have also, like say, the different organelles level. So how that, which organelle system your pathogen is going to utilize or hijack. So those all things will allow you to interact, like if you have the different specific levels for the different organelle system. So, and then, like say, means you try to go with the different FRET-based assays, means FRET-based assay has, like say, limitation that when the two spectral overlapping are in the close distance of, like say, less than 10 nanometers, then only will try to have to consider, like say, means the two interacting entities are pretty close enough, like they are interacting. Otherwise, you're not going to take into consideration. So definitely, despite of, like say, means that's a different aspects, but definitely if we take into account, like say, the different dyes and those things are there. Many of the dyes, like say, even like say, for them, mitochondrial membrane potential based dyes are there, but they all like say depend on the fixation. And I believe like say, although I'm not an expert, but what I have like say, and certain depending upon the type of the fixation or fixative which you are going to use, they will like say affect your mitochondrial membrane potential, and then your respective dyes are not going to label them properly. You will get a false positive. So those all things like even the dyes are there, like say fluorescent dyes are there. So different dyes and different like say the fluorophore molecule of fluorophore analog is going to perform in different conditions, specific conditions. So we should be like say very precise when selecting the type and nature of the type of the fluorophore and a kind of a experiment. But if you and even like say the same dye which you're going to like say consider for the fixed cells, so the simple immunofluorescence may not work out when you're going for the life cell imaging dynamic because of the flow depleting nature, and these all properties get disturbed with the fluorophore molecules. So that's where like say how to like say in context like say it has been challenging to probe the interorganelle labeling of the it has been challenging for decades, and people have been trying to develop like say the different tools and a probe. So because membranes is like say a minimal system which can be probed like say in a natural in a natural how minimum we can affect like say the membrane. That should be our membrane, and here in membrane I mean like say not only the plasma membrane but also like say the organelle membranes. You know what minimum way you can try to affect your like say the organelle membrane or the lipids or the proteins, whatever the conjugate we are going to utilize. That should be very important when selecting or designing of the probe membranes. So to label the different organelle, means like say at the intercellular level, different approaches are being emerging, and like say the click chemistry approach is a one which is being developed like say recently, where people have been trying to label with the different, we know like say our lipids are phospholipids, or the membranes are going to be constituted by the phospholipids, and they have a head group and they have a, like say the acyl chain groups, and these depends like say where we put the fluorophore tag, whether on the head group or whether on the tail group, or whether like say, even on the acyl chain group they have like say the kink like structure with a double bond structure. So that makes like say because if you incorporate like a bulky fluorophore group, that will like affect your membrane. As I mentioned, like so you have to the fluorophore should not have an impact on the natural behavior of the lipids or the proteins. So if you put like say I'm taking example the phospholipids, if you try to put a kind of a, like say fluorophore tag on the hard bulky, and if the diffusion dynamics are going to affect it, so it's not going to like say be the true fluorophore as compared to the labeled and the unlabeled counterparts. So because of the steric hindrance imposed by the fluorophore tag which you label them. And then like say these, similarly the kind of a nature, as I mentioned, like say means where all things, it will also means if you put the fluorophore tag on the head group or on the like say tail group, it will affect like say because of the perturbation, the location, dynamics, and the molecular interactions are going to be affected. So the people have been evolving with the click chemistry, where they try to put like say the functional group in the aromatic triple bond over here, and what they try to do is like say the alkyne bearing derivatives are being used these days, and which can efficiently and effectively label the lipids or the sterols in the in situ cellular environment, at the cellular level itself. And that can be used both for the live cell imaging as well as for the fixed cell imaging of the intracellular organelles. So I'm first talking about the organelle, I'm not talking about the communication. I'm first talking like say how you can approach to label the organelles. That's the first thing. Interaction will come after this. So and that's where like say people have been using these derivatives, and this can be derivatized by synthesize the phospho analogs, being like PC analogs and those things. You can try to design those things, and then these are going to be like say once you allow to get impermeate into the cells, they will uniformly label the both the fixed as well as the live cells, and then application of the alkyne is going to react with the like say propagaryl moiety, which will react with the azidofluorophore, and then you can undergo with the, like, say, different click chemistry based approach, either with the copper click chemistry based approach, or the, like, say, the other one is, like, say, the one we will be discussing with the OCR based approaches. We come to my subsequent slide. So different click chemistry based approach allow to specifically label the specific organelle. These are the one of the recent development which are, like, say, emerging in the field. So definitely it means that chemistry should be very sound when selecting and applying this kind of an approach other than the microscopy. So, and as I mentioned, like, say, means the OCR, the cyclooctane group is required, which are going to, like, say, they are, like, say, less toxic as compared to the copper chemistry based approach, and where you can label, like, say, the different P or PC based phospholipids, and then it's going to be you can image with a different kind of a microscopy. And in addition to that, like, say, there are different other approaches also going to be there. The photoactive lipids are there, cage lipids are there. We will not go into details of that because just because of time constraint, we are not going to cover all of them. So, and even other than the cage-based approach, the photoswitchable lipids are there. Means you try to have, like, say, the lipid tool, photoswitchable tools, like you tag them with the fluorophore, and then you activate it with the lights, and that will allow the change of the conformation. And this changes conformation will allow to get converted, like, say, the phospholipids from the cis to trans isomerization, and your fluorophore will be able to get expressed, and you can, like, image it in the life conditions. So we'll try to discuss in one of the, like, say, a recent paper where the click chemistry based approach has been used to label the ER, because what we say is, like, say, the phosphatidylcholine is the one. PC is the one which is going to present all over the membranes, but utilizing this as a tool, like, means this group they have developed, like, say, OCR based approach to label the ER membrane as well as the mitochondrial membranes. And what they do here is, like, they selectively design to approach the fluorescence labeling and the fluorescence of both the ER and the mitochondria, where they undergo the process of metabolic incorporation of your acetylcholine, which is the label, and then they are going to be utilized by the whole cell system, and wherever there is a PC molecules, the phospholipid, they are going to label that undescribed. So it's here, and now they just try to incorporate your the moiety, the OCR moiety, which they have designed is specific for the targeting to the specific organelle, be it for the ER, be it for the mitochondria. So what here they have done is, like, say, they have designed the one of the molecule is it's on the next one. So you can see it's this OCR, which they have mentioned as a one. It has a fluorophore known as a rhabdol, and that DAPCO, which is going to interact with your alkyne group, which is already you have impermeated with, which is going to be presented by the your acetylcholine group. So once they are going to be there, your cells are going to be labeled, the incorporated cells are going to be labeled with the acetylcholines. And when you allow the fluorophore molecule, the OCR molecule, the alkyne group is going to interact with your wherever and this fluorophore is going to be specific for your particular organelle, and this this strain alkyne group is going to interact with your acido group, and then it will label your particular organelle. That's where the specificity, and this is the important, and the beauty of the system is, like, say, the designing of this particular OCR molecule. That's where the chemistry lies. So because in general, if you try to metabolically incorporate it, it's going to label all the PCs present in the whole cell system. But it's the beauty of the system that to design this particular OCR molecule, and here which is going to be specific for the ER Golgi, I would say. So which is going to, like, say, being activated, and means you just label them, and then they are going to be specific by the click chemistry based approach. They just try to specifically label, and it's, we will see it. And what they do see when they label the cells, the one is like say, I mean, say the OCR molecule which has been designed for the ER and Golgi. So it specifically labels the ER Golgi network, and that is going to be also being contrasted with the different dyes which are being targeting to the ER. And they merge the images. They beautifully see that they are going to specifically label, and means they've also shown that this OCR molecule, which is the number one, the PC1, is not going to label the other cell type, means in these conditions. So it's determined that it's specific for the ER, and they also show that even if you don't apply, like say the cells which has not been treated with your like acetylcholine group, they don't take the, they are not going to be labeled by the specific ER Golgi OCR. So which determine the specificity, or means the reaction, means they are going to target only, and when if the cells are being metabolically being labeled with the with your specific acetyl groups. So, and here it's like say targeted with the PC molecules, the phosphatidylcholine. So that's where the one for the ER, and here they have designed, you mean see that we know, like say the rhodamine is the one molecule which is going to label the your mitochondria. It's well known. So they have designed like based on the mitochondrial membrane potential, I believe. So here they have used the to design the mitochondria OCR molecule targeting specifically to the mitochondria by utilizing a derivatized or analog of the rhodamine and formulating it to form like say tetraethyl rhodamine. And this goes and target and is labeled only to the 2PC. It goes the same way the cells has been incubated with your like say first of all with your acetylcholine group, and now rather than applying for the OCR1, which was being specific for the ER level, now here they just try to apply the OCR2 molecule, which is going to target only your like the mitochondria. And means the contrasting has been done with the rhodamine 1, 2, 3, which is also going to target your, and they have been super merged, and it's beautifully lies like to show you that yes, it specifically labels your mitochondria. And when the super resolution microscopy images were being done, quantification was being done, it was observed like say means here you can see means the one is with the 2PC and the other is with the mitochondrial GF. It will label the matrix also, and the mitochondria as such as like say the outer membrane and the inner membrane and the clustery structure. You see like means it beautifully labeled the 2PC, the label 2PC with the click chemistry. You can see the intricacy of the like the sensitivity of this assay, means of this probe, that it can label even like say the inner mitochondrial membrane crusties. So essentially what I mean to say that it can label both outer membrane as well as your inner membranes. So that defines like say means the PCs and this OCR2 is highly specific for your mitochondria. And now in the lower panel, they just try to label the cells. I think probably the HeLa cells they have taken here. So now sequentially they have labeled with both the PC1 means the OCR molecule 1 and OCR molecule 2. And then they have like say taken with the contrasting with the membrane fluorophore. And then when they try to merge, the specific dyes are means specific OCR molecules label only the one they have been targeted to label. Means the 2PC is going to label with your only with your mitochondrial membranes, and the 1PC is going to label only with your ER membranes. So that it means the specificity and the authenticity of the probe which has been developed to label at the intra-organelle means at the intercellular organelle level. So now means the PC has been shown that means whether such an organelle like helps the organelle interaction, helps in like say how this dynamics helps in the like say the trafficking of the molecule. Means do they try to establish a contact, and if that's the case, can this fluorophore molecules maybe utilized to probe the dynamics of the molecules in a given cellular context? So they just try to monitor the trafficking of the PC labeled from the ER Golgi to the mitochondria. the intraorganelle by the utilizing live cell imaging. What they do is like say they label the cells, the same HeLa cells. They label the cells with the one PC, and then they counterstain, which is going to specifically label the ER, and these ER counterstain, which is well known. They beautifully co-localize, and the beautifully co-localize. The co-localization has been ascertained by the Pearson coefficient, which is of the order of 0.84, which is very good, a very good interaction. But as I mentioned, like say means over the period of time, like say means by one PC, it's what do you mean? Yeah, by 330 minutes, what they do see the intensity of your PC goes down, gradually goes down. So, which is trying to define, like say means where is the flux moving ahead. So at the 0 minute, it's instantaneously, yes, they are pretty high. Means here the imaging has been done in the way, like say for the superposition, that both are in the same background. Both are on the same background. Whatever you see, it's like say the way above intensity. You see here 0 means much of a less is here, but intensity wise, like mean they have negated, subtracted to bring them at the same level of intensity. Then only you can compare that, like means whether the flux is of the flux of the PC flux is going to which level. So to ascertain the dynamics of the PC flow from the ER potentially to the other organelle, what they do observe, like say by 330 minutes, which is like say way almost like say more than 5 hours, they do see, yes, the fluorescence intensity goes down for the PC. It means it is being trafficked. Where does the traffic? They just try to measure at the 0 time point when they label the PC. It's not there. Means by 330 they beautifully co-localize with the PC marker as well as PC, as they do localize with the lysosomal marker as well as with the PC. Similarly, with the within like say as early as like say within an hour, they just beautifully co-localize with the mitochondria, and also like say means in the 2 hours they are being able to co-localize it with the same with the plasma membrane. So it means from the lipids, they helps in like say to maintain the crosstalk with the at the intraorganelle level. So the organelles, they do crosstalk and they are being assisted. Means I won't say that it's not mediated by the proteins, but at least what here, like say the PC labeling is ascertaining that yes, it is being mediated by the lipid, which is like allowing the flux to move from the ER to the different organelle system. And as early as like say in the mitochondria, you are able to see that the flux is being flowing for the PC flux is being flowing from like say from the ER Golgi network to the mitochondria in less than almost in an hour, but like to the plasma membrane or for the lysosomal membrane, it taking more time. So, but one thing is pretty evident: the organelles, they do crosstalk with each other, and here does do reflect like say what is the flux flow. What I've been talking about, like say means at the 0 time point, the PC was it's a intensity of the PC with the super resolution microscopy, and over the period of time with the super resolution microscopy and the live cell imaging was done, you see like say by almost close to 6 hours, the intensity goes down. So, and that's what it means in been tracking from the initial level for the ER where they measure it. It's all intensities measured in the ER. Make sure it's all the intensities we have been taken in been measured in the ER. So, but gradually it goes down, which suggests like say the lipid has been lipids which are localized on the ER or the Golgi, they traffic to somewhere else. And that's where like say the organelles try to maintain and they try to communicate with each other. So, and as utilizing the similar approach, this group also beautifully tried to explain like means a very novel conception, like means autophagocytosis, where the means a different mechanism are there, microautophagy or macroautophagies, mitophagies are there. So for the macroautophagy pathways, the autophagosome vesicles are being formed under certain stress conditions, and these helps to initiate the formation of the autophagosomes, which are going to be like say the biliary membrane structures, and they just try to wrap up around like say the misfolded proteins or the pathogens, and they just try to eventually encircle and they fuse at a point at certain time points, and they means they encircle the means by the recruitment of the different autophagy proteins like C3 or ADG5. And a bunch of the proteins are there, and then eventually fuse with the lysosome with a low pH, the hydrolysis, and they just try to degrade the misfolded of the pathogens. So that's way means that's a and I feel it has been a conception that what is the source of this membrane? Yeah, mitochondria, the plasma membrane, for the early nucleation event in the autophagosomal. And that's a one thing, the source of the membrane. And membrane it can be a protein or it can be a lipid. It can be at the protein mediated or it can be lipid. But whatever the literature has been supported, it has been supported, it can be from the ER, it can be from the mitochondria, it can be, but all they have been reported based on the fluorophore labeling of the protein molecules. And what this group has been doing is they have been trying to label it with the lipid molecule, fluorescent labeling of the lipid, the PC, the derivatized OCR derivatized lipids, which are going to highly specific and target only the ER molecules, or maybe similar, the different OCR molecules, which are going to label the mitochondrial molecules. And what they do see here, when they bring the cells, they label the cells with the similar approach what they discussed before, and they just label the cells, and then they counter stain with the LC3. And as earlier means in the early initial stage, these autophagosomes, they appear as the puncta-like structures, and they will try to colocalize, fairly colocalizes at the very beginning stage. And when the cells were under stress condition for the longer period of time, the puncta which appeared for the the autophagosome, which was being marked by the LC3 by the recruitment of these membranes, the PC also beautifully colocalizes, very beautiful. And even the super resolution microscopy, when they do the line scan for both the autophagosome and for the line scanning done and they try to see the means the superimposing, the line scan beautifully colocalizes and with both the LC3B as well as with the PC, and we suggest that the traffic for the ER means the early nucleation or the membrane which is going to get involved, the formation of this or nucleation of this autophagosome is initiated by the ER membranes. So the PC is the one which is being contributed to the formation of this early nucleation event. So the protein. So now they just wanted to, okay, it's by the, it is by the, you mean by the phospholipids or phosphatidylcholine, which is being contributed by the ER, but has it some role from the protein perspective? So what they do here is they label the cells with the PC, what they did here. Similarly, they label the ER retention proteins, the KDEL with the signal sequence for that. And then they also label with the ER tracker. And what they gradually see over the period of time in the 3 years, where they see gradually they see that the intensity for the PC is gradually increasing from the 0 hour to 3 hours. And the puncta is colocalized as here, the puncta, they do very well colocalize with the LC3B receptor. But for the proteins which are being labeled for the, the ER retention proteins, the different KDL marker protein, they don't grow up. So it suggests that the early nucleation events, even the the autophagosome formation, is initiated by the contribution from the ER membrane. ER membrane lipids are the early initiator, early nucleation event to bring the the wrap up the autophagosome formation. So now comes a question, it's a conundrum in the field, means the PC transfer, means the membrane transfer from the ER Golgi to the autophagosomes. What the route they take, what the pathway they take. Are the lipids going directly transferred from the autophagosome to the ER Golgi? That's the one hypothesis. And the other is are the lipids from the ER Golgi get transferred for the traffic to the separate organelles and then translocate back to the AP? Because initially it was being done means from the protein-based studies that both the ER, mitochondria, plasma membrane, all three contributes to the formation of these autophagosomes. So for this, what they do here is they label the cells with the PC as they have been doing, the PC, what I mean, the OCR molecule, which are going to a specific level. And then they also try to label with the ATG5 proteins, which are like, say, the ER autophagosomal marker. And then they follow up the super resolution by live cell imaging. Then they follow up to try to see that when the puncta is going to appear for the autophagosome formation by the, what they do see, like, say, as early as less than the autophagosome puncta appear pretty early, but followed by, like, say, means the PC molecules, they co-localize fairly by the appearance by almost close to, if you see, like, say, dynamics here is, like, say, the imaging, and the right panel is the dynamics, how the two puncta both for, like, say, the PC as well as for the ETG5, the co-localization and the trafficking aspect is here in the right panel. So that's what they try to see, that how the dynamics is taking place for both the ATG5 and the PC co-localized puncta. And they do see, like, say, early the ATG5 appeared, like, say, as early as here, they do see by the within 22 seconds. That's fine, but here still they are not able to see the PC puncta, means PC is not being able to. So that's the one thing. So, and then they recruit, after the ATG5 is recruited, they recruit, like, say, the PC by as early as by the 66 seconds. And the moment the PC is going to be recruited by the 66 seconds, they eventually follow up the same tracking, means whatever time point they beautifully co-localize with the ATG5, which suggests, like, means yes, means they are the one, means the contributing, the ER to autophagosomal trafficking is being regulated by the ER membrane components or the lipid components. That's a take-home message, I would say, for the vesicle generation. So, and they this group also very well contributed that it's not by the proteins, but it is mediated by the lipid mediated process. Oops. So now we have what we try to cover here is, like, how to level the different organelles. Means definitely, like, there is a spectrum of the approaches to level the different organelles with the, depending on the type of experiment which you are going to perform. But eventually, at some points, these organelles, they just try to crosstalk with each other. And what do you mean by the crosstalk is, means, like, say, the two opposing membranes, they get close to each other. And this organelle communication, be it, like, say, on the right panel, you see, like, say, the organelles have been talking the plasma membrane with the nuclei, ER with the plasma membrane. ER means the organelle ER has been extending itself closer to the plasma membrane. And there are the different, you see, like, different proteins, specific proteins have been localized to these specific organelle. So we will come to it in, like, say, follow-up slides. Then the ER mitochondria contacts, and then ER Golgi apparatus. I mean, they also do to contact, and then as I mentioned, we had discussed, like, autophagy, they also interact with the different other earlier stage with autophagosome formation by for the different organelles, and even the vacuole, like, means for the certain, like, say, vesiculation, and as well as for the osmoregulatory steps, like, say, the vacuoles also contract with the nuclei. So these interorganelle communication depends, like, say, this can be, the communication can be at the vesicular trapping, means the vesicles need to be shedded off, like, say, means generated from some of any of the organelles. And usually, like, say, means if we go with the TGN pathways, or, like, say, the other pathway, it can be by the generated by the Golgi or from the endosome, and the other is, like, say, if it's an endocytosis or by the exocytosis, the vesicles, they just try to, like, say, communicate with the one organ to the organ, or maybe, like, say, out of the cells also. So that's a one way of interacting with at the interorganelle level. Means the vesicles are being released from one organelle, and then they just try to merge and fuse with the other follow-up organelle where they are being specifically designed to go. And the other is, like, emerging concept for the last 1 decade is a membrane contact sites. And such an organelle, like, say, intracellular compartment, means why they do either vesicle trafficking or the membrane to perform, like, and support, like, say, function of the organelle, and they transmit the signals and allow the exchange of the materials. So, and the membrane contact sites, so thus are known as the a very close apposition, a very close contact of the two organelles when they are in a close vicinity. And they should be of the order, vicinity means of the order of like say less than 30 nanometers. We will try to see like say how do we define like say the membrane contact site. So, and this establishment of the contact sites helps like say for the intercellular signaling aspects, the organelle trafficking aspect, inheritance, and they are like say highly regulated. We will see like say means they are highly regulated, means it's not only the membranes which are going to be like say coming in contact, specific proteins and specific lipids are going to be co-localized at these structures. And this is a kind of like say evolving cell biology field at the intracellular organelle level. So how do you define like say the two membrane contact sites? And the two membrane contact sites are like say, although the two membranes should come close to each other, but at least they will be typically like less than or of the order of 30 nanometers. Then only we will say like say, okay, these are like say the two membrane contact sites. The membranes do not fuse here. It's unlike like say the typical fusion and fission, which we really like say covered in the follow-up subsequent lectures. They do not fuse. However, the proteins, they may help, like means a kind of emerging hypothesis or emerging like say protein extended synaptotagmin. We will come back to it. Extended synaptotagmin, they can help like say to traffic the lipids by the process of fusion. So this has been very well proved by the Will Prince from the NIDDK NIH. Although he was the one to propose this one of the concepts, like say membranes do not fuse, but here like say, since the protein is mediating, they have a, like say the amphipathic helical structure, and they allow the trafficking of the lipids when the two membranes are pretty close enough at the order of 30 nanometers. They allow the trafficking of the lipids from the one intermembrane organelle to the other intermembrane organelles. And then specific proteins and lipids are get enriched at the membrane contact site. That's very important. And then like means the formation of such a membrane contact site helps in like say the composition of at least to support the benefit of the two, means at least one of the two organelles. Means one of the function is going to be affected by the interaction of the two organelles. So that's important. So how do they interact? So they are going to interact with the help of the proteins, and the protein or the group of the proteins, maybe one protein or maybe the group of the complex proteins. And they help to allow the bind or like say interact with the two opposing membranes at the contact sites. And we, in the common terms, we say the tethers, membrane tethering. And membrane tethering is like say, means it's involved in all the process of the intracellular vesicle trafficking, whether it be the vesicle trafficking events, or here what we are trying to focus right now, it's a membrane contact sites. And what we do see is like say the bunch of the different listed proteins which have been involved over the period of like say, over the period of like say the evolution, means a different organelle system, means a model system, I would say, be it in a yeast or be it in a mammalian. You see like say the different subset of the proteins has been like characterized or being identified like say to facilitate the maintenance of like say the membrane contact site with the support of these tether molecules. And tether molecules here, what we are saying is like say the different specific proteins which are going to be involved. And in the mammals, like say oristim, it's like say specifically regulating the calcium, intercellular calcium depot. It's like say very specific and highly regulated. And similarly, you see like say ER mitochondrial membrane, MFN molecules, they do fuse, but they also act as a tether as well. They help in like say the fusion of like say, if I'm not wrong, for the mitochondrial outer membrane, but they itself like say in the homotypic manner they interact. Homotypic manner they interact, and they allow like say to support like say the ER mitochondria, the VTAC channel with the IPR3, and these are homolog protein and the other means the fish and the BAP3 and the complexes, the ISAM complexes, they act as a tether, and they also support like say during the fission process. We will come back to it. We will see how to do the support. So similarly, like say ER have like say a contact, established contact with the endosome, lysosome, vacuole, or the multivesicular bodies with the different proteins or subset of the proteins. So it will take like say, means if you go with the one by one all proteins, means we will not be able to cover means in due course of time. But and similarly, you see like say ER establishes contact with the Golgi, the phagosome, the ER, and then with like say your mitochondria with the plasma membrane. So a bunch of all the, even like say the cell within itself, cell organelle within itself, not cell, organelle within itself. ER means the crystal, means the organelle. It's Golgi has like say the internal shaping of the membranes, and they just try to go with like say the protein, grass protein. Similarly, the outer mitochondrial membrane, the inner mitochondria, they also have like say tether with a different tethering protein. So these all proteins like say means they help to support. And if this tether structures or means the formation of the contact sites are being compromised in certain disease conditions, and that affects the functionality of the means what we see. It's like say a pyramid model, which will come back to it. It means it is at the level of like say organelle, and then it's a cellular level, and then at like say the organ level, and then at cell. So that's how like means it's try to grow up. It's a bottom pyramid model, something like that. So, but the evolving concept, as I say, that still it is unclear that these proteins are the group of the proteins are the genuine tethers, which are like say essential to maintain or function the contact site, but are not necessary to maintain the contacts. So these are the kind of an evolving concept, and like say means for the last 1 decade, people have been trying to characterize the different set of the proteins, but how to determine the functionality means you can go with the co-crystallization studies. Whatever the study has been done is mostly by the life cell imaging in those aspects to determine like say, okay, the proteins are there, and like say the different approaches will come back to it. But now the field is evolving, like say how you can co-crystallize, how you can co-purify both the proteins and the functional to determine the functionality of the means of the different subset of the proteins which may act as a tetramolecules. So similarly, the one subset of an structure, what has been determines like say means in the case of like say well characterized first in like say the tetramolecules in the case of the yeast, and where the IST, the MSP, and the CS2 has been shown to establish the ER plasma membrane contact in the wild type. Where you will see that the blue is like say the ER extending closer to the plasma membrane, and they are establishing contact, and this is facilitated by your IST molecules, MSP, which is SCS2, and the TCB, which is C2 domain. C2 domain is the one which is going to have like say the affinity for the calcium. C2 domain bind, C2 domain bind the PIP2. They're going to bind with the PIP2. So these are well characterized in the yeast, and the homologue of these proteins are going to be the extended synaptotagmin. For the TCBs, it's going to be this extended synaptotagmin, which is having a C2 domain, and they bind with the PIP2 localized on the plasma membranes. Imagine that now these subsequent 3 sets of the proteins are going to be replaced by the extended synaptotagmin over here, TMO16, and the VAP proteins in the mammalian cell system, mammalian plasma membrane contact site. So these are all the one like say, and what they do is find like say if there's like say the abrupt in the case of like say means if the tether is not maintained, if the compromised expression of the tether, if like say the proper contact sites are not established, as you see in the right panel here, there's an increase in the UPR signaling pathway. So kind of a stress signaling pathway. You can go more detail about it, or maybe and they also led to like say the accumulation of the PI4P accumulation on the membranes. So that's and this will like say help like say to develop a certain other disease consequences. And similarly, the tethering dynamics has also been routed to explain in like say in here in the muscles, I believe, and where they are like say the how the receptor induced function of the establishing the contact sites helps in like say the very elegant approach of like say maintaining the calcium signaling pathways. And when there is like say when the two membranes, where the ER and the plasma membranes, they approach to the close proximity of like say 20 nanometers or less than that, with the rush of the means the intercellular calcium signal depot, because ER is going to the one like say the maintain as a calcium depot. And what happens like say when the ER is they are going to be released, it allows to interact with the PIP2 which are going to be localized on the plasma membranes, and those PIP2s are going to be hydrolyzed to the IP3 and the DAG, and then here like say the IP3 is going to be like say routed and means and they allow to recruit the extended synaptotagmin to the site, which is like say closer to the ER. Means you see, when they are going to be hydrolyzed, they recruit the green extended synaptotagmin, the one I talked here, extended synaptotagmin, the TCP in the existing in your yeast cells. These extended synaptotagmin, they establish the contact with the C2 domain. And C2 domains are like, say, as I told you, that they are going to bind with the PIP2. The C2 domain has a, like, say, the functional affinity for the PIP2. And they just try to hold the plasma membrane and the ER membrane contact sites. And as a result, like, say, they have, like, say, they recruit a bunch of the other proteins to facilitate, like, say, and maintain the ER plasma membrane junction for the functional activity of the in the muscle cells to maintain the calcium signaling. And one thing we just try to see at the gross membrane level, which I think our next speaker will be covering in the follow-up lectures, like means it's still not yet clear for the membrane contact size. Yes, we know, like say the two membranes, they just come close to each other. PIP2, with what rate the PIP2s are get, like say, routed or recruited, how they are localized. We know about, like say, the calcium proteins are there in it. But in addition to the, what are the accessory proteins? What are the dynamics or lipid changes taking place in just, like say, these contact regions? These are all things we don't know yet. Means further literature or further research do needs, like say, to explore in this direction. Like, because, like say, the PIP2 is the one thing, but you see, like say, it's like say the recruitment of the bunch of the proteins. It's not one thing. Once the extended synaptotagmins, they just try to interact with the molecules. They recruit, like say, the bunch of the proteins. And what are the kind of a lipid changes taking place? What are the new molecules? How they are? Means possibly, like say, the change in the lipid, like say, the emergence of your phosphatidylserine, or means other anionic lipids. Means this all needs to be characterized, but and how the functional complex, the co-crystal structure, it's that's the one thing which is limited, because if you go at, like say, the gross cellular level, like mean these entities existing, like say, very it's a highly dynamic. Means the rate of, like say, the formation and disruption is pretty fast. And means I was not able to show you that video, but the dynamics is very fast. So and these all gets compromised during the disease condition also. So means proper, like say, capture of this particular stage is very important to ascertain the function of this tether as well as the contact sites. So they also helps, like say, means contacts helps in the exchange of the lipids and allow the cells to modulate the lipid composition of RNA independent to that of the vesicle trafficking pathways. Organelle receive the lipids from the membrane biogenesis by the non-vesicular trafficking, which takes place at the contact site. These are, like say, we have already covered these things. And lipid transfer, the MCs regulate the lipid metabolism by channelizing the lipids to or away from the enzymes in different compartments. So it means two different compartments. One is going to benefit it, but one is going to be supported, or both are going to support each other, but one is going to be differently, like say, benefited during this course of, like say, process. And then, like say, the different protein, the specific proteins are going to be involved in, like say, the to mediate the exchange of the lipids during the trafficking pathways. So here, like say, the is going to be facilitated means because the lipids, they facilitates, like say, the means there are, like say, in like say, normal process, like say, the different protein lipid trafficking proteins are there, but here, like say, the SERT proteins, they initially, like say, the LTP, which we say the lipid transfer protein, they function by shuttling between the membranes at the contact sites. They shuttle between that, and they just try to hold the proteins together while they are simultaneously bound to the other membranes. Some LTP has, like say, ability to exchange one lipid for the other, like means what do here they just try to, sorry, it's so initially the LTP here, like say, the SERT proteins, which are going to be when the PIP2 is going to be recruited at the contact sites, they just try to recruit, like say, your LTP protein, the SERT, and then this allowed to transfer the phospholipids from the one membrane to the other membrane. You see, the red is going to be transferred, routed to the upper membrane. So that's why they try to shuttle the lipids from the one to the other. And they just also help in, like, say, the holding the two membranes together. So definitely these are going to be, like, say, means what the nature of this, like, say, because they are going to be pretty hydrophilic in nature, because they just try to, like, say, hold the head group region rather than, like, say, the tail region, which is if it's going to be the tail region, this entirety should be, like, say, the hydrophobic in nature. So many of the proteins which are being characterized to be act as a lipid transport proteins are, like, the OSPV proteins, the third protein which transfer the ceramides, and these ceramides are going to play a number of the process in the number of the disease conditions, which we others say, like, say, the NI2 proteins. And the other possibility is, like, say, unlike the LTP pathways, we can also support, like, say, by the exchange by forming the hydrophobic chain. That's what, as I told you, like, say, means the one in the earlier LTP, they were trying to, they're trying to have the channel, like, say, the pocket for the hydrophobic head group region, at least in the case of, like, say, your SERT protein or the LTP proteins. But here, like, say, it is allowing the hydrophobic channel, like, means hydrophobic channel, so where the lipids are being routed to take path through this protein. They hold the protein, the same protein is going to hold, bridge, they act as a tether, and they have a channel, which allows, like, say, to traffic the lipids from one membrane to the other side of the membranes. And that has been very well shown in, like, say, ER mitochondrial complexes, and then synaptotagmin, like, means proteins, SMP proteins has been shown, which have a channel to transfer the cholesterol from the one membrane to the other membrane. And the other thing I also mentioned, like, say, means the certain proteins have also been shown to undergo the process and mediate them by the hemifusion pathways. And what is hemifusion is, like, say, unlike means if you try to make a membrane phospholipids, so phospholipids can form a planar lipid. It's going to be the single membrane, like a monolayer of the membranes. But our plasma membrane is a bilayer membranes, isn't it? So what happens, like, we will cover these in details, like, say, when we go with the fusion and efficient pathways, but just try to give you, like, say, since the terminology is the hemifusion stage. So when the two membranes approach to each other, what happens, like, say, the outer bilayer mixes, the mixing of the outer bilayer, because since you are bending the membrane, they're coming to closer and they are, like, say, undergoing the process of fusion. It's going to be, like, say, means highly hydrophobic in nature, and the membrane has to pay the price for that. And this price is being paid, being paid off by facilitated by the presence of the different lipids or by the different proteins. So and outer bilayer mixing is being mediated. The stage when the outer bilayer mixing means of the two membranes which are closely opposing, when they merge and together, the stage is known as, like, hemifusion. And when this is hemifusion is followed by the pore mixing, means now you see, like, say, when the two membranes, just take, like, say, the two membranes are here, and two means they are being just bent together, and this outer bilayer just mixes here over here. Only now that what is left here is, like, say, which is not well mixed, is inner bilayer membranes. If this also get mixed, the proper, like, say, the connection is being established within the two volume of the cells. So here, like, say, you see this outer bilayer mixing is being facilitated by the certain proteins. And here, recently, the very elegant group from the Yale, I think Peter Di Camali, they just established the synaptotagmin. They facilitate, like, say, the lipid transport by the synaptotagmin. They help in, like, say, theta, as well as they supporting by the hemifusion. But mechanistically, how? It's not being yet reported. So I think those are the kind of future possibilities to study in this direction. And I already mentioned, like, say, means how they are going to maintain the specific calcium function. So the ER plasma membrane has a very important role in regulating your intercellular calcium levels in the cellular systems. So when the calcium concentration in the ER is very low, and it triggers the calcium entry into the cytosol and the ER from the outside of the cell. So that allows, and that helps, like say, means by the SOCE receptor molecules. So these are the ones which are sitting over here. So that will allow it to help, like say, entry of the calcium molecule getting inside into it. And then the plasma membrane responsible for the entry is, like say, they will try to recruit the ORI protein. ORI is how they are going to recruit the ORI and the stem protein. They are the one which I think we have seen before. They are both together. They act as, like say, or tetramolecules, but they act as a tetramolecules, and they act as, like say, to regulate the intercellular calcium, maintain the intercellular calcium concentration at the contact sites. So what they do see is what they do report, like say, the plasma membrane responsible for the ORI and the sense they sense that the calcium concentration in the ER is low. The stem protein, they are going to sense here that it's going to be low, and then they are going to be recruited at the, like say, the contact site regions, and as a result, activation of this stem protein will allow to shift them being evenly distributed on the ER to form the, like say, the complex between the ER or stem 1 and the ORI1 proteins, and that will sustain this complex formation to maintain the inward flux of the calciums and to maintain the proper concentration of the calcium within the ER depot. So they also helps in, like say, the membrane remodeling, and this very well described in case of, like say, the mitochondrial fission process. And we won't go into biophysics because how the dynamic and those things are involved here, but at least the take-home message is that the ER and the mitochondria, it's a very dynamic, and this mitochondria, they undergo the they're in the continuous stage of the fusion and the fission process. It's highly regulated by the recruitment of the bunch of the proteins, and what decides, like say, the site of the place where the mitochondria is going to undergo the process of fission, and that's going to be determined by the action of the formins. Like say, the first, like say, your ER mitochondria are tethered by this ER and mitochondria. This means going to be by the tether protein arms. ERMES means, in check what's the full name, and that's one of the proteins. And then this is, like say, ERMS is in the case of the yeast, and the homolog protein in the in your mammalian system is named to be something different. You have to cross check with that. And then this tethering of the ER and the mitochondria will allow to wrap up the ER around your mitochondria. So you see, like say, I mean, this is determining the site, the recruitment of the ERMS protein. This is determining the site, and the first to get wrapped, the ER is going to wrap up your mitochondria, outer mitochondrial membrane, and then as such, ER encircles your mitochondria, so that this determines a particular site where they are going to, and then now they recruit the different formins. The actin and the formins are going to be recruited, and as a result, now they recruit, like say, the DRP1 as a fission proteins in the case of the yeast, and then this makes the fission or initiate the fission process by the constriction. So they just try to, like say, and this stoichiometry is going to be very important. To what an extent, to what the number, how many number of these, like say, molecules are going to be. I mean, these all the things are, like say, very important from the biophysical structural biology perspective, that how these are going to be determined, means to what rate, like say, the DRP actions are going to be performed. But at least what determines, like say, first, what determines the site of the fission is, like say, ER is going to wrap up your mitochondrial membranes, recruitment of the actin, the formin to will drive, recruit, like say, your fission proteins, and then the mitochondria undergo the process of fission. So what we do see is, like say, means MCS or contact sites have a number of applications in the different, like they perform a bunch of the functions in the different cell physiological system, or the intraorganelle physiological system, not a cell physiological system. But what are the different approaches has been used? Means, you know, a classical perspective, like say, people have been using a membrane core fractionation approach, But a limitation has been like say means they just try to get the population or the pool of the organelle was not like say pretty pure in one way. So they were already have an impurity of that. While the purification process means or the isolation of the different organelles means they were like say uh, have a purification or means they were impurity with the other organelle as well. And then they try to have like say the chromium precipitation where they try to see like say how the protein level they just try to interact or the two when they're at close proximity, they try to capture that particular state and then undergo the process of chromium precipitation if they are to going to like say means interact at that time point the for the isolated membrane fraction or means the contact size with the organelle. And then that rationally they have been using like say in the literature like some electron microscopy and electron microscopy as we know like say means it's a fairly good enough, but it allows the compartment and to achieve a better resolution, I would say, of the order of nanometers and angstrom. And over the electron microscopy, I would say a better achievement has been done by the cryo electron. At least it will try to like say, since it's a membrane, all these are membrane, it will try to have a less impact on the membrane. So because even like say if you perform electron microscopy means your membranes are going to be like say means at a rupture or means a processing or handling itself like say bring some sort of an artifact. So the cryo-EM has a much more advantages and a good part is like say you can achieve a better resolution of the order of like say even the angstrom level like say your resolution which try to will determine the function of your like say the contact sites. I mean this will help you. Those are the approaches at least like say both the proteins or the intact contact sites can be co-crystallization is still not reported to my understanding. At least I have not come across the literature, but at least the crime structure has been reported. But the greatest weakness of this either the EM or the crime approach is like say means for both the process you need to fix the cells, but at least the cryo-EM retain like so you freeze down the cells when you whatever like say the structure means they will maintain that integrity and there will be less artifact on the cell or the organelle membrane level. And then comes like say people have been using to use the genetically encoded fluorescent fusion proteins and which allows like say the dynamic probing of the fluorophore for the live cell or the contact sites with a high molecular specificity. And the BIF-C function, the biomolecular interaction, what we do is like say means people try to label like the fluorophore tag on one of the like say a protein which is going to be sitting on one of the membranes, say for an ER, and one protein which is going to be sitting on the mitochondrial membrane. And if they are going to be in a close apposition, like means and close apposition means you will see like say the change in the like say the fluorescence in the increase in the fluorescence intensity, and that allows you to like say means the organelles are interacting. So, but care should be taken while using these, like say, means to prevent the spectral overlap. Means while the process of emission is taking place, with the different fluorophore tag which you are trying to apply or encode or tag with the different organelle, they should not overlap because then you will have like say the overlap of the spectral imaging, and then will like say your output or outcome it will be tough to interpret. And an emerging field is like say means how the different organelles are being interacting, and that will like to have a bigger picture like say the means to define like say the organelle interactome. Means the way like say the simple example is like say the FACS has been using to characterize like say the subset of the different seven or eight like say the max I think, yeah. So a similar approach has been done recently with the Jennifer Lippincott from the an SCHD, where she has been using it to able to define, like, say, the multispectral imaging and the computational studies to define the organelle interactome with the different subset of organelles using the different fluorophore molecules. But whatever kind of an imaging approach you are going to take, best considered tool, it's like, say, dissolve, like, say, means the fluorophore interaction will tell you, like, say, to build up the hypothesis, yes, they are probably interacting, but you need to prove it them with the different biochemical approaches, different structural approaches to prove that, like, say, what is the functional entities of means how what kind of a sort of a complex they do exist in form. So a very beautiful and elegant approach has been developed the same by the same with Jennifer Lippincott, where they have been trying to hello, hello, am I audible?
Yes, sir.
Yes, sir.
There are some messages there. I can't. Slides are not seen. Is it so? Can you see the slides or not?
Yes, sir. Slides are seeing, but since too long, only one slide is been seen: lipid transport protein and lipid exchange at MCS site. We are seeing this slide. Slide is not appears to be changing.
I'm just why didn't you have interrupted? Okay. It says my slides are shared. I don't know what's an issue. I will just try to share again. Then you could have interrupted. Like means. Are you able to see now?
Yes, sir.
What slide? What's the title for that?
Lysosome positioning and function in different cells.
And what was the earlier slide which you didn't see? What was the earlier slide? What was the last slide which you were able to see? No idea. Means were you able to follow, like say, how they were involved in, like say, the fission and those things which I was talking about, the arms protein? Huh? Have you seen? Have you gone
slide
through?
Number 20 was being seen.
Okay. So in the next slides, like say, I just try to cover, like say, okay, means here, like say, I just told, like say, in addition to, like say, the transfer proteins, there are channel proteins which are, I will just go fast, like say, they help in, like say, to mediate. They have, like say, the channel hydrophobic channel which allow the trafficking of the lipids from the one membrane to the other membrane, so that they shuttle the lipids between the two membranes. And they also, and the same protein, they act as, like say, the tether protein as well. And here, like say, means the extended synaptotagmin protein, they have been shown to undergo and mediate the lipid transfer from the one membrane to the other membrane by the process of hemifusion. And hemifusion, I think you will be covering, like say, in a more detail, like say, in the membrane remodeling section, where at least just to give a brief glimpse, like say, for the bilayer membrane, where the two outer layer are going to be, like say, in a close proximity, they are going to merge and they form, like say, the diaphragm-like structure. There is a, like, so when the two membrane get closer to each other, they buckle. The buckling is, in the biophysical term, that term is known as the buckling. And then when the buckling is going to, like say, maintain the contact, then they go to form, like say, diaphragm-like structure. You see, this is a diaphragm-like structure is formed. So this inner bilayer, this outer bilayer, which is now in the merge stage, they have already, like say, merged, but the inner bilayer is not yet merged. And like if a more, like say, the means the job means more pressure or more, like say, deformation is being taking place, this will allow to get, like say, the merge even the inner bilayer membrane. And this job, like say, merging of the outer bilayer membrane or the inner bilayer membrane, is in the different condition and the different situation being facilitated either by the lipids or by the proteins or by the both. And here, like say, means the hemifusion can be also one of the route to transfer or traffic the lipids from one membrane to the other membranes. That's what here. So I will skip this one. Means this is the same thing which I'm trying to see, like how this ER plasma membrane in the muscle cells, they are involved in, like say, the maintaining the calcium homeostasis. And here it was how the ERMS protein in the yeast helps in, like say, your in the, I'm not able to, is my, oops. So how this, like say, the different tethering protein, where the tethering protein helps in, like say, the both the establishing the contact with the ER and the mitochondria, and they also helps in, like say, the fission process of the mitochondria. So initially, like means the ERMS protein, which has been known as a tether protein in the case of the yeast, they are going to be recruited in the mitochondria because they are in a constant continued dynamic phase of the fusion and the fission process. They just get recruited at the site where the fission process has to be taking place. And then they just wrap up around it. And then after wrapping up, the actin and the formin, and this actin and the formin, they just try to generate a high curvature domain, like means regions. means we will get into it like say in the subsequent slides. So, and then like say this recruitment of the formin acting the site of like say the wrap of ER recruits like say the DRP1 protein in the case of the yeast, which is the fission proteins, and analogous to your dynamine, and then they just try to help in like say the fission of the mitochondria. So basically that like say means the ER and MCS contact site determines the fission site in the case of like say your mitochondria. So here I have discussed, I think I have discussed about, do I need to repeat it, or for the membrane co-fractionation, I've already gone through it. So means the different approaches like say have been utilized to undergo and to study the membrane contact sites, like membrane co-fractionation step, where like say from the cellular system, people have been trying to, or different group have been identified to isolate the different organelle, like say in a contact form, but they were usually having a kind of an impurity with that. So that limited the utilize such a approach, or at least if they utilize, if they were purified, it was like say not able to maintain the contact sites. And then like say the co-immunoprecipitation, where you try to like probe the protein at a interaction level. And the electron microscopy has been classically used to study at the lower resolution, means resolution level of the nanometers and angstrom level, but they also have like say prevent because their cells are the membranes need to be fixed. They impose an artifact on the membranes, and this led to like say usually this led to like say the membrane rupture and those things, and which may allow not to like say have the two membranes in a close contact. You may lose that. Those are the kind of an artifact, and that's why like say now the approach is going with the cryo-EM. The moment you try to establish, or you believe like say you have the contacts, you isolated those, you just freeze it down by the cryo-EM approach, and then go, and that will like to at least retain the structural complexity of your contact site entities which are going to be there. So that's where at least you try to overcome with the limitation of like say, although being cells are fixed, but at least they will retain their native structure. And now means using the different super resolution microscopy and the live cell imaging, people have been using the genetically encoded fluorescent fusion proteins, and which allow the dynamic event to follow the dynamic event of the current organelle contact site. So what I would say, like say, means not a single approach is going to work out, but at least it's like say the multifunctional approaches to determine to build up the hypothesis, as well as to explain like say means the functional entirety of such structures. So a recent group a study has been shown like say means by the BIFC approaches and like say the bifluorescence microbe based approaches, they are trying to show like say means in a one organelle type, they try to express the proteins tagged with the subset of the protein which are going to be localized on the one organelle, and the other half of the proteins with on the other target proteins. And then when they when they are like say membranes are pretty close in contact, when their contact sites established, they both the fluorophore, they are going to complement each other. And as a result of complementation, you can able to see the fluorescence, and then you can ascertain that yes, the membranes are in like say fairly close enough and they are in a contact. And that's how like say means people are trying to deliver much more work has been done in context to the ER mapping, like means ER and a mitochondria, but it's still like say how the other organelles are lysosome, peroxisomes, and different vacuole, multivesicular bodies, how they just try to interact, how the interact with the different organelle systems in a dynamic manner. It's yet to be ascertained, and that's how like say the people are progressing into identify and establish organelle interactome studies. And have to run fast, I think. So this is an elegant approach developed by the Jennifer Lippincott, where, as I mentioned, like say, means it's a very dynamic process, and it's very tough to capture those contact site regions. So, but utilizing the property of the phase behavior of the membrane, like the phase behavior of the membrane is like say something where the membranes, they can undergo in the process of a different ordered and a disordered state. So what I mean by that is a liquid ordered and a disordered state. And this can be mediated by the composition of the lipids in the membrane or the nature of the proteins which will be present. And that allow, like say, to membranes to achieve, like say, different order and disorder. And that gives a rationale to form, like say, the nano domain like structure formation in the membranes. So I think I know, like say, whether if you have gone through with that or not, but this will be more covered in by the our speaker Saurav Halda when he is going to study about the membrane biology, means biophysics of the membrane biology. But imagine, like say, the ordered and a disordered stage are the very important player in, like say, the nature of the phase behavior, like how the nanodomain like structure, and the nanodomain structure something is going to establish a give the concept building for the lipid rafts. And if those are the sites, like say, means if you are able to, and they were able to beautifully capture those ordered and disordered state by simple approach of, like say, the blebbing. How can you bleb? Either by the, like say, chemical approaches of blebbing, the cells shut off the vessel. You can just, the cells are in the tension stage. They are highly tense. And likewise, the membranes. If I talk about the cells, the membranes are in a highly tense stage. If you relax the membrane tension, the cells will, like say, bleb off. Like they just try to shut off the vesicles, the blebbing. And it can be either induced by the chemicals, which are going to form, like say, giant plasma membrane vesicles. And there, like say, you can easily define, like say, mean that a different membrane to level, like say, the ordered and the disordered region of the, like say, your lipids. And if the membranes are already in a contact, one ER here in the plasma membrane contact, or vice versa, means in the different organelle system, they have been able to capture, like say, means ordered, means not only, like say, the contact site, but also in presence of, like say, the liquid ordered and a disordered phase. And this ordered and disordered phase is really helpful in determining and further studying of, like say, the means structural perspective, from the signaling perspective, how they recruit the different bunch of the proteins at the contact site region. Because it's very well defined, like say, in case of, like say, the cell plasma membrane limitation, but it's, like say, the kind of a very emerging concept in the field of, like say, the intercellular contact regions. And what they do, like say, they just simply allow the cells to go a hypertonic shock. You just dilute a PBS with the, like say, 4-time dilute. It's going to be, like say, and you apply it, the cells will bleb off. That's a hypertonic. That's the beauty of, like say, the very simple approach of, like say, approaching the cell biology. And they just rationally capture, like say, the different organelles, and then they try to, like say, further determine by the different approaches. So it's a very elegant approach of, like say, tool to, like say, capture such contact site regions. So as I mentioned, like say, means so we have covered, like say, the different aspects of your contact sites, but in a umbrella model, we see, like say, means if there's a defect, means the organelle level interaction helps to control, like say, especially in a time and a space, to regulate the protein functions, and the protein functions will regulate, like say, the different functions, whether the rapid trafficking, maintaining the calcium signaling pathways, membrane trafficking events, which eventually regulates the process at the cellular level. And that will eventually regulate the process at the organ level, means, and then eventually impact your pathology. So that's where, like say, the field has been evolving, like say, to study the organelle interaction and the dynamics with the different approaches, what we discussed right now. And just shedding a light on that, lysosome has been one of the aspects where it has been shown to be involved in, like say, the number of the event, means in the neuronal synapse region, means when the two synapses, or the axon and the two neurons are in a close contact, means they just build up the connections. And the presynaptic region and the postsynaptic regions, the intracellular vesicle trafficking, means we will cover it. They just try to release, like say, the vesicles, and means where the vesicle recycling process is taking place, and autosophagosome, they just try to, like say, undergo the fusion with the lysosome to utilize, or means degrade the uh, the the vesicles or the means the organelles, as well as like say they are going to be endocytosis as well. And this flux in the release of the endocytic vesicles from the presynaptic region and the uptake of like say the vesicles or the means by the postsynaptic, it's going to be like say maintained by the like say not only like say the dynamic but the positioning of this lysosome, because that's very important also where those positions are being utilized, because and it's even sometimes we will try to discuss in how the fusion is going to be regulated. That means if the nuclei they get fused, but how they are being located, that determines like say the pathological index. And similar like say and like say the cells undergo the process of phagocytosis, they just try to engulf and like say they undergo the process of fusion with the lysosome and they just try to kill and dissolve the pathogens. Similarly, in the TCR signaling pathways, where the immunological synapses are being established, the LAT proteins which are going to be recruited at like say the TCR regions, they just undergo the process of the endocytosis, and then like say to maintain that receptor density, they also undergo the process of like say the fusion with the lysosome and then they degrade it. And similarly, like say with the here, they just try to show in the case of like say the autophagy when the nutrient imbalance is taking place. So you see, like say lysosome is at least one of the function involved, like say degradation activity in the bunch of the physiological condition. And if that is going to be affected, this dynamics is going to be affected at the different synapse regions. Means when the two membranes are in a close, when the two cells are in close contact, they will affect, like say, to develop some sort of a number of the disease conditions. Means and they led to the accumulation of, like say, the different disease, the cholesterol, the sphingomyelins, and which has been very well defined in case of the different lysosomal storage disorders. So I won't go into details of it right now. So for a while, I can just skip because we are running short of time. So here, like say, means for the membrane dynamics and the contact sites have been studied a lot over the 1 decade, but people are also trying to target the different this dynamic event and like say the phenotypic in the different disease condition. And here, like say the bunch of like say the clinical trials are going on where people have been trying to target like say different tethering proteins or the proteins which are involved in like say the contact site establishment or the fission process. The different approaches are being used which are in the clinical trial to see the what will be the outcome in the different like the different disease conditions. And mostly targeting, we see like say mostly targeting the mitochondrial proteins which are going to establish and which are involved in like say fusion and the fission process. So next part was like say to cover like say the trafficking event, the membrane trafficking, and the route which is going to be like say means it's a how the cargo molecules are going to be uptaken, means the kind of a delivery pathways, how they are being trafficked, the vesicles, how they are being going to be trafficked. It's a different pathways of the cargo delivery system within our like say cell system. On the membrane level means that depending on the direction of the flow will help us to design like say whether the pathway is going to be endocytosis or an exocytosis. And these vesicles are being generated either from plasma membrane by the different methods, by the clathrin-mediated endocytosis pathways, by the phagocytosis pathways, by the micropinocytosis pathways. And then there are like say the different cavioles which are like say rich in the sterols and the sphingomyelins. They are going to form like say the coated and non-coated vesicles, and they are just going to be internalized. And these internalized particles, the pits, they are undergo either mean depending on the nature of the cargo molecules which are going to be endocytosed. They just ride or go with the retrograde flow of the vesicles are routed to undergo and fuse with your early endosomal, and then during the cycle, either going to cycle back or to undergo the late endosomal, and then they try to degrade those then. And then the next is like say your exocytosis, which is being reflected by the blue arrow. So means The vesicles which are being generated inside the cellular system from the different organelle level, be it like say the ER membrane vesicles, the and traffic to the ER to the Golgi is what is mediated by the COP2. However, like say when they are going to be like say generated from the Golgi and revert back to the ER, it's by the COP1 vesicles. These are COP, the clathrin, COP, and these are like say basically the coat proteins. We'll come back to. So these are the coat proteins, and then they just try to like say either ER to Golgi or from the Golgi to ER. So one is mediated by like say the COP2, and the other is by the like say, and since like say the your ER is a major organelle where the like say the protein synthesis and the other activities are going to take place. They are being shuttled and they have been routed in the form like say the cargo is being loaded in the vesicles, and then they are being shuttled back to the traffic to the Golgi, and then from the Golgi, from the trans-Golgi network, cis-Golgi network to the trans-Golgi network, they are being routed from the Golgi cisterna, and then through like say they are being released and they undergo the process early. They can undergo the and fuse with the early endosome and then recycle back to the membrane. And then when they are going to be released outside the cells, the process is known as your exocytosis. So when it's going to be something, the vesicles which are generated on the membranes getting inside, endocytosis, and they're being routed outside, released, it's exocytosis. So pathogens, different like say the receptor signaling pathways, means they get activated by the endocytosis pathways, hormone release of the hormones, the cytokines, these all are going to be regulated by the exocytosis. And bunch of the different proteins, they assist in the formation of the different subset of like say your, and the very primary thing what we do notice is like say the shaping of your membranes. You see, like say means whether be it your endocytosis pathway, whatever the endocytosis pathways are going to be taken, they are going to take like say means they are going to shape up the membranes. The membrane forms the vesicles, the bulging of the means either inward or the means when they are going to be released outside, like say then the vesicles are generated from one like say your different organelle, it can be like say ER or the Golgi, and then they are going to be routed and then fuse back. Finally, eventually they are going to fuse back to the membranes. So it's a highly regulated process. And these are going to be supported by the bunch of like say the proteins, and now I will say not only the protein, it's also by the lipids as well. It's also by the lipids as well. So what we try to see like say in all these events, any of the endocytosis pathways, the membrane has to bend. You see they have formed the vesicles, and to bend the membrane has to pay the cost for that. And this cost is being paid by the composition of the lipids or the nature of the lipids which are present at the site where the endocytosis or the exocytosis is taking place, or being facilitated by the proteins. This cost is being paid. But in general, like say, we see like say, not only the body, but they have a different shape as well. You see, they have a different shape. Even the different cell also have a different shape. The neuronal cells, or be it like say the muscle cells, they all have a different. How it is shaping? It's all by the curvature. The membrane curvature is the one thing which define like say your what kind of a shape the cell or the organ or the intercellular pathways or vesicles are going to take place. And in a two-dimensional system, like say you can define the curvature by the radii, but it's for the cell system you visualize that. For any sphere, if it's a circle, you can have a one radii, and that will define like say one circle. But if it's going to be in a three-dimensional space and a time, you will have basically your like say the two radii, one imaginary line passing through this and the imaginary line passing, which like say, and if you try to like say extend this thing, and if they meet the two line where they meet, this is going to define like say the principal radii for the two. And for any shape, any 3D shape, you need to have like say the two radii, two principal radii, and that will define the two curvature, the C1 and the C2. That's a thumb rule. And the combination of this curvature, the C1 and the C2, which is the inverse of your radii, principal radii, is going to define like say the total curvature and the Gaussian curvature. And depending upon that, the cell is going to take a different shape, be it like say either cell itself or it's in the endomembrane system itself. So the basic shapes which the cells can take is like say the planar, where the like say your curvature is defined by the monolayer or it's by the bilayer, where all the parameters are zero, and that's been beautifully observed in the membrane layer, which has been beautifully like say means covered up with the Catherine lattice. And this beautiful image was being like say by the John Husser, and I was lucky to follow him. John Husser, well, is my next lab. So we used to, he's one of the pioneer who has like say, design a very elegant, like say the endocytosis structures. And the next structure is like say the simple, these are the basic membrane shapes. And depending upon that, if your C2 value is zero, but rest are like say positive, then it's going to have like say your cylindrical shape, the tube-like structure, where you see like say dynamic coating is taking place. The membrane is a tubular and the dynamics are coated. These are like say the image from the John Husser. And here is like say the other four out of the four, like the saddle shaped structure, when they like say when the pinching off of your endocytosis, even when they like say the neck bud, like say you try to create a GPM, like say the giant unilamellar vesicle structure, and try to coat it with the different proteins, janine especially, and then you see how the membrane budding is going to take place. So depending on the nature and the composition of the lipids, they are going to adopt like say the different shape. And the sphere, it means you simply try to, all are going to be, the sphere is going to be all, you're trying to in vitro reconstitution of the Clathrin cages, where all the like say the parameters are in the positive direction. So that means the curvature is the one which is going to define like say what kind of a shape the membranes are going to take place. So, but those are the basic things. But in general, in principle, in the cell biology, the curvature is going to be like say adapted by the facilitated by the lipid composition or the presence of the proteins. Means if you have a like say the lipids with a big head group and a short tail group, that will try to, and it's the nature of the lipids will like say help in like say how they partition into the biliary membrane, how they just try to like say fit inside like say one next to each other, and that will give like say the bulging effect, the bending effect. And that helps like say to take helps like say assist to take the membrane to take a proper shape. Your like say if it's a conical lipids, it's like say the big head group and the short tail group, that will like say means the notion you see the membranes are going to bulge out, and that is going to define like say the positive curvature. If something is going to the flat, imagine the visualize like say a flat flat membrane, a biliary membrane. If it bulges out, it's a positive curvature. Inverted cone shaped lipid, phosphatidylethanoamine, diacylglycerol, cardiolipins, these all are going to be have like say the short head group, but a bigger tail group. That will like say inverted cone, and that will give us a negative curvature. That will impart a negative curvature. The cylindrical, the PC, the phosphatidylserine, phosphatidylcholine, these all give a shape membrane like say the cylindrical shapes. So, but in general, like say means you see the composition of these lipids defined like say means the membrane as such is a sea of a flat, but the changes taking place by the composition of these may have a, like a local effect. It cannot be like say all everywhere. It can have a local effect. So means the nature of like say whether it's a positive curvature, the negative curvature as such per se can be a highly localized event. And then like say means the other than the lipids, the proteins may also facilitate. And the shape of the proteins and the extent of like say the proteins may impart the similar means like say the proteins whose like say the extended terminal means exposed transmembrane region is like say the bigger, and they just try to like aggregate. You will see that they generate a positive. And similarly, like say if the shape of the proteins are going to be like say the inverted cone shape, they can impart a negative curvature. And apart from that, like the proteins may also get like say the proteins, they may also help in like say the amphipathic in helix molecules. There are certain proteins which can just if they impermeate, even in like say the in the monolayer membranes, the helix region is highly amphipathic in most of like say the bar protein. The bar proteins, we come back to they like say whenever the membrane has to get the shape, these are the curvature generating, curvature sensing proteins. They are curvature generating. So at least the amphipathic helices, and amphipathic helices, like say, mostly found in like say in many of the viral proteins also, and those when they just get inserted into the one of the monolayer bilayer membrane, you see, not inside. They just try to like say fit inside, like say fit between the two head groups. And the extent, like say how the bigger they are, they can impart like say the gene to induce like say the local positive curvature. And that's being facilitated by the bunch of like alpha-syn nucleonapsins and those like say bar proteins. Similarly, the C2 domain, which is like say the affinity for the PIP2, and has we have already mentioned like say the synaptotymine or the DOC2. They just try to fit in like say outer bilayer membrane, and they just impart like say the induce a curvature. Then the loop insertion, the proteins they just get inside to impermeate into the bilayer membranes. Here they just try to like say, but doesn't across like say the head group region of the other bilayer region. So the caveolins, which we see in the caveolin-mediated pathway of the endocytosis. The bar proteins, these are like say they have a banana-shaped structure, and they just try to have an affinity for like say the curved region. They just sit on that, and they just try to bend the membranes. They just sense the curvature, and they just bend the membrane. And these are the like say one of the very curvature. The bar proteins are also known as the curvature generating CGPs, the curvature generating proteins. And these are like say they assist in the formation to take membranes at different shapes. And that's how like say means we will come to it like say endocytosis, which it's like say in like say when it's dynamic mediated, which itself is like say the coat proteins which helps to take, but one is like say the fission process, but one is like say the dynamic independent, which are going to be facilitated more by the bar proteins. And even the bar proteins, like whether if they help to like say shape up the proteins inside the cells or outside the cells. So that also like say define like say in which directions the membranes will like say fold back. Whether if it's an N bar, they will go inside, F bar is going to be outside. So and these depending on the nature of the proteins and the lipids, this can be of the means earlier they were like say the nanoscopic structure, but now if the curvature changes at the level of the microscopic level, so those are being facilitated by the different coat proteins, by the scaffolding protein. They just try to recruit a bunch of the different proteins, and like as I mentioned, like say the COP1, which is going to be like say from the Golgi to the ER, and the COP2 from the ER to the Golgi. You see the different subset of the coat proteins are there. Coat proteins, they help, like say, help to shape up the coat, help to take the shape, a different shape. The clathrin media, and beautifully designed in the case of the endocytosis, they just wrap up around like say the vesicles with the help of like say the different coat of means the adapter proteins, the AP1, 2, and 3, and that helps to take the different shape. The protein scaffolding is assisting in the formation of the membrane bending, because it's the very first step involved in like say any of the like say the shaping process of the membrane. And as I mentioned, like say in other than the scaffolding, the bar protein itself, they have assist in like say the how they are being arranged, like say that will also help like say to constrict the membranes accordingly, if they are like say side to side, or if they are arranged like say from one top down bottom approaches. And apart from like say the proteins, like say the cytoskeleton proteins, like say that we do have seen like say the phallopodia like structure, the tubule like structure, the lamellopodia. These all primarily they do involve like say the actin and the accessory proteins to bend the membranes, and basically they are like say enriched to form the highly curved structure, and which are like say rich in the membrane tension. Highly curved structure with a rich in the membrane tension. They are going to localize their protein of interest, and they are going to like, but the tubular shaped structure, you see that the IBAR proteins, they will try to allow you to make, see, give a shape inside. They will just sit inside this and inside the tubular membranes. They're not outside, mind it, they are not outside. And then they will just try to ping it along with like say in the ATP dependent manner. They just try to shape up the membranes to form the phallopodia like structure. Similarly, your dynamine and the myosin. Myosin is the one protein which is like say involved in the shaping of the membranes, and other than that, like say in whenever there is a like say the ruffle formation, the different cellular structure, they recruit the different protein, the VASP and the wave complexes. It's a you can go ahead with like say what are the signaling components involved in that. So we see like say means whether it's endocytic pathways or the exocytosis pathways, the very first event is called the curvature generation. And we know like say this curvature generation can be facilitated by the proteins or by the lipids. So and here like what we're trying to see like say means these are highly curved structure rich in the membrane tension. The different channel proteins which we see, they are sometimes sitting on like say, they are enriched in those sodium potassium, these potassium channels, which are like involved in a calcium channel. They are like say enriched in these highly curved, highly tense region. And they means the curvature operates of like say 10 to 30 nanometers here. And they may support like say either receptor clustering or receptor exclusion, depending on the nature of the like say the physiological behavior what we are trying to or study. And then they may have like say the role in like say the fission process, as well as you see the coatapone proteins or the clathrin proteins, they recruit like say the dynamic proteins at the point when they have to pinch off from the membranes. And accordingly, like when they have to go for the fusion process, they will try to the different protein which have an amphipathic helix-like nature. They will try to insert in the two membranes, and they just bring the two membranes in the close position to allow them form the undergo the process of fusion here. And then, like say the C2 domains, when they help, like say in the release of the vesicles when they undergo the process of the V snare and TCR fusion process. So curvature is very important. These are the vesicles. They are now designed to fuse with the target membranes, and they are going to be supported by the helix-like structure, the 6-bundle helix-like structure, the snare complexes. It's a one protein, but it's a bunch of like say the multiple proteins. And they eventually what they do is like say these helix-like bundle structure, they will try to pull the, I told like say you have to pay the cost of the membrane bending. And under different conditions and different situations, they are being facilitated either by the lipids or by the proteins. And here, like say the snare complex, they form a helix-like bundle structure, and they're allowed to bring the two membranes together. And this is what's the buckling or the docking of the like say the vesicles. They dock each other, hemifusion followed by the content mixing, and now the vesicle is going to release outside. Whatever the cargo molecules are sitting here, they will release. And then, like say when the pinching off of like say your the vesicles while the endocytosis, it's a low curvature region, like say means they recruit like say when the endocytic pathways are going to take place. The synaptogen, not synaptotagon. Synaptogen is in low concentration. When the PIP2 is going to be hydrolyzed, because PIP2 till the time PIP2 is present, endocytosis will continue to take place. But for the release of the vesicles from the membrane, the PIP2 concentration has to go down. And when they are going to be hydrolyzed, in the presence of the ARF GDP cycle, the synaptogenin concentration goes high. And then they try to like say constrict the membranes. They just try to constrict the membrane to release it, to generate, and constriction of the membrane will try to generate a very high curvature rich region, and that will allow the fission of the budding of like say your membranes from the plasma membrane to the endosomal CCP, like clathrin-mediated endocytosis process. So, but the proteins and the lipids, they can just change the lipid itself can maintain like say curvature. The protein itself can maintain the curvature. But emerging hypothesis has been like say is they can function in like say in a coherence. They can like say means they can crosstalk with each other to facilitate the process of like say the membrane curvature generation. And that's what in here it has been shown like say how the AT1 and the clathrin coated vesicles from the trans-Golgi network facilitates the process of your curvature generation in the yeast cells. So DSRP2 is like say the flipase, and a flipase, it helps like say to flip your phosphatidylserine from the in ATP dependent manner. It's minded they're like say the different set of the proteins which help like say in the shuttling of the anionic lipids across the biliary membranes. Scramblers are the one which are like say ATP independent process, but here it's a flipase, it's ATP dependent process, which here the ATP is going to be like with a RFGF based process, where in the E cell, this allow the the facilitate the phosphatidylate inositol recruitment, and as a result, the this flipase is going to recruit your ARF protein when the RGFP is bound. As a result, the ATP dependent process, these GTPs are going to be hydrolyzed from, and as a result, they are allowing the exposure of the phosphatidylserine from the inside to the outside in an ATP independent manner, mind it, in an ATP dependent manner. So what happens, means this protein facilitates the recruitment of the lipids from the inside bilayer membranes to the outside bilayer membranes. See, they are going to be enriched over here. The blue dots, they are the PC, PS composition. The PS is the one which is in the blue, and now they are going to be sitting outside, most of them. And as a result, when the PS is going to be recruited on the outside of the bilayer membrane, they will recruit a bunch of the different proteins, because PS is a kind of they act as a formation of a nanodomain, and PS is known to recruit a bunch of the proteins. It's a behavior of this protein, the lipid, phospholipid, that they recruit a bunch of the protein which form the signaling components. And all the proteins are going to be sitting here. And as a result, here, based on the functionality, whether modifying lipid composition membrane molecule or the protein domain, which is going to assist in either they are the white collar worker or the blue collar workers, depending on what nature of the whether lipid is involved or the proteins are involved or both are involved, they couple together to induce the curvature change, because PS as such here is going to being recruited outside. And I would argue here, because PS being a cylindrical nature, ideally it should not bring a curvature change. But when there's a kink in the PS molecules, that has been reported, kink in the the SL chain molecule, then they may potentially impart the change in the membrane curvature. So it's a PS whether recruit the protein, for sure, yes, but bringing a change in the curvature, that is a kind of a debatable issue yet. Means that need to be further, means still it's some people buy it, some people don't buy it in the field. I mean, in the membrane biology field. So means some people do buy it, some people don't buy it. So here, the very first step I show you, same here, bending of the membranes to take the different shapes, either by the budding process or the sorting process or by the fission process. So, and these are going to be facilitated by the the lipid composition, the actin networks, the protein crowding, and the and the curvature rich domains. When there is a budding process, the escort mediated the protein helps in the viral budding process. So what comes next is we see it can be clathrin dependent process or it can be clathrin independent process, depending upon the nature of the proteins and the pathway and the cargo molecules which are going to be taken inside the cell. So usually clathrin dependent pathways are going to be EGFR endocytosis pathway, low density cholesterol pathways, and the caveolin pathways, and which led to eventually means getting back, they will helps to form the cage-like structure facilitated by the clathrin ring axis and the AP2 adapter proteins, and they forms the coated vesicles. And caveoli are the rich in macrodomain structure, which are forming these on a bilayer membrane. They are enriched in the your cholesterol and the sphingomyelins, and they are the small vesicles which are rich in the caveolins and the cavins, and they facilitate to form the bulb or the vesicles, which are clathrin independent. You see, there's no clathrin, and these are clathrin independent, and then they help in as a stress sensor, and they have a prominent role in the ECM remodeling. We will see in the other subsequent slides. And then is the clathrin independent carriers, where mostly facilitated by the GPI link receptors and facilitated by the different bar proteins. And because the bar proteins, they help in rather than the vesiculated, they will allow you to form the tubular structure. So we see, like say, there are the bunch of the proteins which are listed proteins which are being listed in, like say, either clathrin dependent pathway, clathrin independent pathway, cavalry mediated endocytosis, the different subset of the proteins which are involved, and like say what can be the mechanism, how they may be helping or regulating the, like say, the endocytic events in the different conditions. So in general, like say, a typical pathway for the endocytosis is going to involve, like say, the recruitment of the clathrin triskeleton, which is going to be recruited on your vesicles, which they coat the membranes, and the wrapping of the neck is going to be involved, like say, the dynamine-like structure, and apart from that, the site is going to determine by the composition. The site is going to be determined by the PIP2. When the PIP2 concentration is high, the vesicles will continue to grow and form, and the PIP2 is going to be hydrolyzed by the synaptogenin, as I mentioned before. They're going to be, like say, now the dynamine is going to function, and they will constrict your neck region, and then allow the pinching of the DNAs. Pinching of the vesicles. So, and how they are going to be, like say, recruited means how the clathrin gets recruited to the plasma membrane. So as it's going to be triggered by the PIP2 accumulation, the PIP2 gets accumulated on the membranes, and as a result of the enzymatic activity of the PI4 kinase activities, and this as a result, and not only by the PI4 kinase, PI4-5 kinase. It's a group of, like say, the PIP2 is a group of, like say, 13, and 13 lipids. We'll see in one of the slides, and they are going to be, like say, they are going to be interchangeable in the, like say, and then different stoichiometry to depending on, like say, what all hydroxyl groups are going to be phosphorylated. So now the PIP2 is going to be recruited, and as a result, that once the PIP2 is going to be enriched on the membranes, those are going to define, like say, the site of the place where the endocytic events will be going to be initiated. And now they recruit, like say, the bunch of the adapter proteins, which we say as an AP2 complex, A2P, AP2, and now the AP2 is going to also recruit, like say, the different accessory proteins with the CLAPS proteins, the actin myosin, and then initiate the process. I mean, at least these proteins are going to be recruited at the very first level to initiate, like say, the your formation of this clathrin recruitment. And now the bar proteins are going to be recruited on the membranes, and which will allow to form the bulging of the membrane. And depending on the, like say, side of the PIP2 sites, they are going to, like say, means recruit bar proteins, and these bar proteins amplifycin or the endophilins are going to give shape to the membrane. They bend the membranes, and accordingly they will allow the deformation in the membranes to form the clathrin coated bits. And as a result of this, the membrane is going to be invaginated, and as a recruitment of the process, the other accessory proteins which has been recruited, they will wrap up around, like say, on the emerging vesicles, the vesicles which is continuing to growing, and different proteins are being, like say, recruited. The receptor proteins, when the carpet loading is going to be there, the receptor protein as ligand binding will initiate, like say, the membrane deformation by the recruitment of the membrane, and the triskeleton composition will try to wrap up around the membranes, and they continuously try to grow. And when the as I mentioned, like say, when the PIP2 level grows down here, now the pinching of the DNA, pinching of the vesicles will take place. So it's the same thing which I mentioned, like say, PIP2 is initially the PIP2 concentration is high, but they are going to be wrapped up with the different molecules as we discussed before. But once the PIP2 concentration is going to be low, it's going to be hydrolyzed, then allowed to dissociate, like say, that means the adapter proteins, when they just get dissociated, and then they gradually allow the dynamine to constrict the neck regions. And this stoichiometry of the dynamine is very important, and has been biophysically ascertained by the pulling approaches of the membranes, which I think Dr. Sara will discuss in the subsequent lecture. These vesicles, like say, the bar proteins, these are, like say, these are topology of the membranes, and they dimerize to cleave your vesicles. I'm not going just because of rush of the time, like say, we are running out of time. I'm just trying to be fast now. But if you have any problem, you just email me. I will try to get back to you with different questions if you have. So the uncoating is going to involve, like say, means the different protein. The means when there's an uncoating of it recruits the oxygen proteins, and this when bind to the clathrin core, and then they recruit, like say, the heat shock proteins, the ATP, and then as a result they disassemble. They disassemble the triskeleton models, and the vesicles is going to be released. So this is a more affinity for your triskeleton clathrin molecules. As a result, like say, it get continue to dissociate it. But mind it, like say, these all processes are taking place when there is a, like say, the drop in the PIP2 level. So other than the PIP2, like say, the other pathways of the endocytosis are which are clathrin independent pathways. You see, these are, like say, the caveolins, which are found by the caveolin and the cavin proteins, which give shape to the vesicles, which are going to be generated. And importantly, they are going, let's say, this scaffolding protein, they are, like say, rich in, like say, these caveolins, these caveolin regions are rich in, like say, cholesterol in this sphingomyelins. And importantly, this is a hairpin-like structure, the cavern-like structure, the hairpin-like structure, and they are, like say, involved, they are palmitoylated. And that makes, like say, the proteins, and that makes, like say, the shaping of the vesicles, which are enriched in, like say, the cholesterol and sphingomyelin. And such kind of a pathway has been shown to, and this is, like say, your clathrin independent pathway of, like your endocytosis, and has been involved in, like say, the endocytosis of the SP41 and cholera toxin and albumin and many other protein. And other than that, like the caveolin has also have a precise role in the mechanization and the ECM remodeling. So they act as a kind of a stress sensor. When there's, like say, you flatten the membrane, means if there's some sort of an shear stress imposed on the membranes, they get disassembled, and as a result, like say, they just sense the stress. So depending on the nature of, like say, the stress the membrane is facing, they just undergo the process of, like say, they regulate, like say, the membrane tension. So that's the one thing. And it has been shown, like say, the cavin proteins, like say, they are, like say, recruit, like say, the caveoli in the rich region, like say, which are going to be, like say, the row gap pathway, which like to, like say, the recruitment of, like say, the actin-actomycin contraction, which facilitate, like say, the migratory activity of the cancer cells, and helps, like say, in most of the tumor cells, where they modulate, like say, the extracellular ECM modeling. And as a result, when there's a defect in, like say, certain cancers, cancer process, the migratory metastatic nature is going to be compromised. And just because, like say, this actomycin contraction is going to be blocked, and as a result, like say, ECM remodeling is, like say, compromised. So a number of disease has been affected by these, and mostly, like say, the affecting the muscular dystrophy, where the membrane tension, the muscular membrane tension is being compromised, and these are the, like say, the muscular dystrophies, which are the disease and like relevant, like say, the proteins which do affect, like say, the caveoli mislocalization and other addition muscular dystrophic disease, and definitely we discussed for the cancers and. For the COPD disorders also, like say, it has been shown that the caveolin is, like say, compromised in the functioning for the smooth muscle cells and the endothelial smooth muscle cells. And then comes, like say, the other non-endocytic pathways of the endocytosis, which is, like say, the like GEC, and these are, like say, your clathrin independent carriers, and they undergo the process in the GPI anchored and rich compartment. So that's going to be facilitated only by the bar protein and the CLEG and GEC, the clathrin independent carrier, and they are going to be, like say, when they're going to be GPI enriched, they undergo the process of, like say, the tubule formation, and further, like say, exocytosed by the fusion in the retrograde manner. They undergo the process of fusion by the early endosome, and then they have to cycle back. So that's a continuous process being maintained, and helps in, like say, the carbon utilization, the membrane repair process in the dysfolding proteins, which has been reported in, like say, the muscle membrane repair process. And it's also utilized, like say, in the endocytosis of the streptolysin. And next is your ARF1, and ARF1 is, like say, a clathrin independent plasma membrane recycling process regulated by the AF6 protein and is a member of the AR family of the GTPases. And in this pathway, they cycle the inactive GTP to the active GTP bound state and at the plasma membrane and facilitate the internalization of the cargo molecule. And similarly, undergo the process of like say the retrograde manner to recycle back the endosome, fuse with endosome, and then recycle back to the exocytosis. And such process has been involved very well in like say the MSC class 1 molecules like endocytosis CD40, 55 and 59, and the number of cell adhesion proteins like the catenins and the integrins. Oops. Fast endophyllin mediated endophyllin is like say your bar proteins. They just encircle around your membranes, emanating bulging membranes, and then they upon activation of the different specific GPCR by the ligands, they just undergo internalize, and then by the PI3 signaling kinases, they are going to be phosphorylated and then undergo the process of the endocytosis. And as such, down the lane, they undergo the retrograde transfer trafficking, where they are going to be like say fusing with the ER Golgi and then recycle back to the release to the exocytosis, either the early endosome pathways or by the late endosomal pathways. And other than that, like say means a macropinocytosis and is also involved, where it's like say the cell drinking process, but unlike the shaping of the membrane is going to be initiated by the actin-dependent process. And most of like say cell differentiation process, macrophages or the internalization of the EGF and PDF mediated, they undergo the wrap up the solute molecules, and they just form like say the rich bigger vacuole-like structure, which is going to be facilitated by synexin bar proteins, and or they can just go undergo the homotopic fusion one is to other process, and form the like say the macropinosomal structure and undergo, and eventually they fuse with the lysosomal for the degradation of the granules, and then they are being fused back to the membrane. So, and this have like say a lot of physiological implications, the antigen presentations and the pathogen invasion pathways. And the depending on the cargo vesicles which are going to range is they can try to enwrap like say the bigger size of the cargo molecules. And same here is like say the phagocytosis, but rather than cell drinking here, they is being mediated by your binding of the pathogens across with the receptors, and this receptor molecules they recruit like say the actin-myosin ectomyosin contractions, and as a result, they just led to the ectomyosin contraction led to the like say the bending of the membranes, and as a result, from the lateral, from the like say the ectomyosin contraction tried to emanate on both the two other side of the like say the pathogens, and as a result, like say the wrap of the pathogens or like say the cargo molecules from both the side, and eventually they fuse at the sealing end, and then they are going to be like say routed, and after internalization, either they go for the fuse with the phagolysosome for the like degradation, and the lysosome is recycled back. So how the field has been evolving, like say the imaging like different endocytic pathway. Classically, people have been able to resolve and study such an fine vesiculated structure in EM studies, and then fluorescence. But the fluorescence is like say the fluorescence microscopy will just try to flash off like say the lights which are going to appear when the budding or a vesicle is being generated, right? And when it has been more prominent to study with like say today's days with the here the multicolor FM-based endo means they just try to label the different cargo proteins with the different fluorophore molecule tags, and then just try to follow the what the route they are going to, what the route the vesicles are going to take. And people have also tried to like say crystallize the lattice structure of the by the cryo-EM model, which is going to resolve the structure, endocytic structure at the angstrom levels. And now they're just trying to combine, let's say, correlative light sheet microscopy, means where they're trying to see the both the fluorescence, they label the fluorescent probe, and then we also show it with like say your EM structure, and then they are going to be overlapped. And if they co-localize, that means that's going to determine. And this is like say the one of the the recent structure for the last 1 decade people have been trying to follow up. And other than that, like say means in vitro reconstitution of the FR domains. Means if we are trying to able to reconstitute the means how you typically try to generate the model membranes and then try to apply your bar membrane, bar protein to see the functional reconstitution, like how the fission and the the giant plasma membrane vesicles are going to be tubulated by the application of the different bar proteins, and that will help you to understand, like say, how big and how what kind of a shape and how they are going to take a shape, and in a both in the GTP dependent, ATP dependent, and in a non-dependent manner. That will help you, like say, to reconstruct and study the molecules in vitro conditions. And definitely, like say, means how fast, what's the dynamics of the fusion events or the generation of this endocytosis event has been more studied by the super resolution microscopy. And as I mentioned, there's a correlated super resolution microscopy, both the fluorescence microscopy and the electron microscopy. So you combine with, like, so you level the flow, as I mentioned here, like say, here it was like say, not at the super resolution at the confocal level, but now here is like say with advanced super resolution microscopy, where you have, like say, your cryo structure, as well as, like say, super resolution microscopy structure, where you can precisely localize your protein on, like say, the cryo structure, and then you can see the pinpoint, like say, where all, like say, your proteins are sitting within the of the order of, like say, at least down the lane 1 to 2 nano angstrom. So the field is continuously evolving, like say, and to get a better and in-depth insight of this endomembrane trafficking events. So as I mentioned, like say, the exocytosis, I will try to be very fast. I've already taken much time, but exocytosis event, like say, when there is, like say, the secretion of the vesicles from the inside to the outside, and it can be, like say, either constitutive secretory pathway, which deliver the membranes, lipids, and protein of the cells, and there's a, like say, regulatory secretory pathway, pinching of the vesicles, and the vesicles are going to be, like say, it's a regulated, means it's going to be controlled by, like say, a number of the means either secretion of the hormones or neurotransmitters, and there's some sort of signals being involved and outside to inside, and that makes release of vesicles, and then the vesicles are going to be regulated. And we will also get to know, like say, what are different mechanism of, like say, the fusion events. And then there's, like say, the lysosomal pathways, which are going to eventually make to fuse the vesicles which are generated from the lysosomal pathways, and eventually fuse with the outside of the compartmentalized plasma membrane. In the regulated process, you see the mid one, the vesicles which are being generated and are, like say, docked on the membranes, they can fuse in the 3 different manner, and, like say, kiss and run. And it's going to be, like say, your membranes are sitting the brown color is, like say, membrane and the vesicles, and they just approach to each other. They just try to sit around, and then they just like wriggle around. They just fuse, and they just and endocytose back. They just release the content, and they just go back. Then there's a fully collapsed model. The vesicles will fully collapse, and they just merge with the target membrane. And the compound exocytosis, one of the vesicles will fuse with it, and the follow vesicles will try to join with and then they will try to maintain, like say, the connection. And these are, like say, being found, like say, depending on the nature of, like say, the regulated exocytosis, it's being found in a neuronal synapse, endocrine release of, like say, different hormone, exocrine glands. And the beauty of, like say, the system is that for the different model system which you study, the range of the vesicles which are being generated are also different. The vesicle size is going to vary. For the neural synapse, it's of the order of 50 to 100 nanometers. Endocrine is of the 60 to 300 nanometers, and of the exocrine cell line, it's going to be of the order of the micron. So it means the release of the cargo vesicles, and there's also means different in the different model system for the regulated exocytosis. We could have gone more, but we will try to cover possibly the one, like say, how these kiss and run in the fully collapsed model, because it's going to involve, like say, the full fusion pathways, and it's going to be covered more in, like say, the membrane remodeling aspects of our subsequent lecture. So all in all, like say, other than the proteins and the vesicles and the shaping membranes, the PIP2 is the one, like say, the regulatory signaling molecules, and these are, like say, the molecules which have an unsaturated head group, and, like say, your they have a phosphodiester linkage, and of the total, they are, like say, I think 13 different PIP2 exist, and depending on the site, which are hydroxyl sites, which are going to be phosphorylated, in the different permutation and combination, the different PIP2 is going to exist in the cell site and the cell membranes, and facilitated by the different PI3 kinases, how the hydrolysis is maintained by the PGen kinases. So, the regulatory balance of the existence of such a PIP2 molecules is going to drive any like say endocytosis event, and both at like say the plasma membrane levels, and as well as we already discussed the contact trace region. Just imagine, but for the contact, it's not yet precisely explored a lot. So how they are going to be targeted on the membranes, like and the PIP2, like say means which is in an inactive state, when they recruit like say your PIP kinase and effector molecules, they are going to be activated either at the endocytosis level or at the endosomal trafficking levels, or at the like say when they are going to be involved like say in the lysosomal function. So any be it anywhere like say in the membrane traffic like say pathways, PIP2 has to be the initiator. And then when PIP2 at the means they are going to, how they are going to regulate like say your clathrin mediated endocytic pathways, the clathrin PIP2 kinases, when they are like say recruited, means they are going to be enriched by the PIP kinases, and the recruitment, I think we already discussed, they recruit like say different number of the dynamic and adapter proteins to recruit like say the functional reconstitution of the like say your vesicles, and then they recruit like different dynamine and the ARP2 and the other curvature generating CGP proteins to generate like say the vesicles, and as a result the vesicles are going to mature. Now the vesicles need to be released, and to release of the vesicles, the PIP2 is going to be hydrolyzed and for the dephosphorylation process, and they recruit like say the synaptogenin and other proteins, and this allow the dynamine proteins to now get it allow to constrict the neck region more and more and more, and as a result the pinching is going to take place. So for the fusion and fission, like say at least of the order of like say less than 30 nanometers will allow to compress the dynamine, and in the different stoichiometry, we will see it in a subsequent lecture that and this helps like say the constriction of the vesicles to release the endocytic vesicles from the plasma membrane to release. And similarly in like say the exocytosis process, when they help in like say docking the exocytic vesicles, when they approach the plasma membrane where they are going to be released the content, they are going to be form a complex with the exocyst complex, which are going to recruit like say the PIP2, and this is in the case of the yeast model system. And also it do involves like say the monk proteins, they get also recruited and activated by the binding of the PIP2, and they eventually bind with your vesicle molecules, and CHAPS protein, they also get bind with it, and as a result the priming is going to be mediated by the monk protein. But when the snare protein, which are helping in the fusion process, we will see it when the help is going to, it's a multi-protein functional activities for the fusion process. Unlike the viral mediated fusion, which is going to be mediated by the single protein, it's a multi-protein complex machinery where the snare is going to mediate a fusion process, but it do involve like say a number of the accessory proteins which is going to be activated by the PIP2, be it a monk protein, be it a CHAPS protein with a synapsin protein, they are going to be activated. And then after activation, the snare will facilitate, and the formation of the 6 helix bundle will allow to form the fusion of these exocytic vesicles with the membranes. I think this we have already covered. And a number of the like say the disease has been by the defects in like say the signaling pathways of the PIP2, or with the endocytic trafficking or the exocytic pathways, or in the actin remodeling events. They have been involved in like say a number of the pathogenesis disease by the cancer cells, be it in the COPD even, or be in the myopathies, or in like say the different pathogen infections. So PIP2 is like say very crucial for a number of like say the lipid related disease pathways. So with this, I would like to thank you.